So welcome, everyone. We're pleased to first start with our early stage investigator of the day is Li Du. She's a postdoctoral fellow in Satish Pillai's lab at Vitalant. And she's been studying mechanisms of HIV persistence with a particular focus on some immunoregulatory mechanisms that are also linked to HIV reactivation. Galectin-9, and she's actually doing some really interesting work studying a monoclonal antibody targeting this pathway for efforts to assist in the clearance of the HIV reservoir. So we're really excited to have her join and give the ESI presentation of the day. Take it away, Li.
Thank you so much, Peter. I'm going to share my screen. Thank you so much. Good to see you, everyone. I'm a little bit nervous, sorry. Thanks, Safer, for giving me our opportunity to share our work and connect with people in the field. I'm Li. I'm a staff scientist in Dr. Pillai Lab at Vitalant Research Institute. Today, I will be sharing our recent work about modulation of GAL9 to target HIV persistence. So to give some background, as all of you know that the untreated HIV infection is characterized by the high level of viral replication in the plasma of people living with HIV. ART could effectively suppress HIV replication to undetectable levels. However, if ART is interrupted, HIV rapidly resumes. The reason why HIV rebound following ART interruption is because of the HIV latent reservoir. Even with ART, there are several ongoing viral replication may still occur to result the new mutations and the new integrations in the susceptible cells. And moreover, the latently infected cells could persist for years due to the longevity and the capacity of proliferation. Therefore, identifying host factors that modulate the latent reservoir will inform HIV strategies. One of our interests in the study is galectin-9. Galectin-9 belongs to the galectin families, which are the immune modulatory beta-galactoside binding protein that regulates lots of viral pathogenesis. For example, the HIV, the influenza virus, and the HCMV, etc. During the COVID time, our data showed that GAL9 could enhance SARS-CoV-2 replication and immune pathogenesis in the primary airway epithelial cells. Beyond that, GAL9 was also demonstrated to play a dynamic role in the context of HIV infection. Our work lab, along with others, made effort to demonstrate that GAL9 may promote HIV pathogenesis. One recent study showed that one recent study using the HIV-infected humanized mouse model showing that GAL9 could promote HIV RNA and DNA level in the tissues, suggesting GAL9's role in the regulating viral reservoir in vivo. Interestingly, recent study published by the several SAFER lab members showing that the baseline GAL9 was identified as the strongest predictor of the greater subsequent decay of intact HIV, suggesting its dual role in modulating and maintain of the HIV reservoir dynamics. Based on that, our main purpose for this project is to investigate the mechanism underlying the GAL9-mediated HIV reservoir expansion and to access the potential of P4D2, which is an anti-GAL9 monoclonal antibody provided by our collaborator from Cornell University to eliminate the HIV reservoir. As we discussed in the background, that our previous study showed that GAL9 could reactivate HIV latency. In our data, in J-light cells, GAL9 could reactivate HIV expression. However, if we added the p42, the monoclonal antibody, our data showed in a dose-dependent, the p42 could inhibit Gal9-mediated HIV latency reversal in a dose-dependent manner, indicating Gal9's effect on Gal9's antagonist, p42 antagonist, Gal9's effect on HIV latency reversal. Moreover, Gal9 has also been shown to activate the resting CD4 T cells, and our data showed Gal9 could activate the resting CD4 T cells through interacting with the CD45, and of course, added that the p42 could inhibit the activation, indicating the potential therapeutic of p42. Next, to investigate the Gal9's effect on HIV infection, we pretreated the resting CD4 T cells with Gal9 before infecting them with CXCR4 tropic or CCR5 tropic virus. 3 days post-infection, our data showed the increased infection in Gal9-treated cells, regardless of their tryptophan. These data indicate that Gal9 renders resting primary CD4 T cells more permissive to HIV infection. As expected, the p42 could protect CD4 T cells from Gal9-enhanced HIV infection. To investigate the underlying mechanism about how Gal9 enable HIV infection, the SAMH1 is the well-known restriction factor which could inhibit HIV reverse transcription by depleting the DNTP pool. SAMH1's activity is regulated by the phosphorylation and the proteasome degradation. Our data demonstrate that Gal9 could promote SAMH1 phosphorylation and decrease SAMH1 total expression, which inactivate the SAMH1 to enhance HIV infection. As with the previous experiments, the p42 blocks Gal9-mediated SAMH1 deactivation. In summary, our data showed that Gal9 signaling promotes HIV persistence via these several mechanisms, and the p42 anti-Gal9 monoclonal antibody is a promising therapeutic preventing Gal9-mediated immune activation, HIV infection of resting CD4 T cells, and the reactivation of latent virus. As you can see, our work is still ongoing, and there are lots of gaps we need to address. And moving forward, we are going to use the CRISPR activation 293T system to overexpress the Gal9 to mimic the high expression of Gal9 in the people living with HIV. And then we will co-culture the engineered 293T cells with the resting CD4 T cells for several days, and then to measure the T cell activation, SAMH1 deactivation, the susceptibility to HIV infection, and we will perform the single-cell RNA-seq using the 10x genomic platform. Finally, I would like to thank my supervisor, Dr. Pillai, to supporting and directing this project, and all of our lab members. I would like to thank all of the collaborators, and I would like to thank NIH to support our project, and I would like to thank the CIFAR Award to supporting our another project about interfering regulated restriction factor on HIV persistence. Thank you so much. That's all of my presentation. Any questions or suggestions I would like to take? Thank you so much. Yeah, that's all.
That's terrific, Lee. Thanks for an outstanding presentation. We'll open it up to questions. And Lauren, I can't see the people who may be raising their hands. So, but I can ask a question to start with. In some of our own work, looking at the immunologic predictors of mortality in treated HIV, galectin-9 also pops up as a unique predictor of mortality in HIV. It doesn't seem to predict mortality that well in the general population, but does in people with HIV, suggesting there's something about HIV that's really impacting this pathway in important ways. And so I'm really intrigued by what you're finding with the reservoir. Have you thought about ways that intervening on galectin-9 might be used along with other HIV cure approaches? Might it enhance other potential cure strategies when used in combination, do you think?
Yeah, I think so. For example, the Gal9 could reactivate the HIV latency reversal. I think one of another things uh, uh, one of another thing I think we can combine with other probably um uh, LRAs to promote the infected cells goes to the program the cell death or yeah, I don't know if I answered that question.
Okay, great, great. And it looks like Steve has his hand up. Steve, you want to go?
Yeah, I mean, that was a lovely presentation. Thank you. I really was able to follow it, so that's great. So just with Peter, that JID paper with that Gal9 relationship to the reservoir size was pretty dramatic.
Yeah.
And I could never quite get my head around what it meant. Because if it increases transcription, that shouldn't really have an impact on the reservoir during treatment, right? Because treatment will block any virus from replicating, in theory. So it has to be doing something else. And I always wonder, the main driver of the reservoir size seems to be T cell proliferation. Does Gal9 cause T cells to proliferate? Could that be?
Yeah. I haven't shown one of the data is our Gal9 could promote the proliferation of the stem cell like memory CD4 T cells, which is also increased the permissive of the stem cell like memory T cells to HIV infection. And this stem cell like CD4 stem cell like central memory CD4 T cells was demonstrated as negative and self-renewing signaling pathway. So it could be like promote its proliferation of the infected cells. And also, I don't know some theories, but even with the art, there might be still some viral replication may occur in the tissues. We don't know. We don't know if it's true or not, but we need to demonstrate that. Yeah, but
Yeah,
I
that
think
debate.
one of
That
your
debate
paper
still
published
persists.
still
Yeah,
suggested that, right?
yeah, that's complicated. Yeah, that's
Yeah.
still an argument being made. But thank you.
Thank you so much.